Amanda, can you hear us?
Sincere apologies that I'm not there in person. Not everybody can control Lufthansa's ability to keep their planes running. So I'm here by phone instead. I hope you can hear me well. We've been talking a lot today about how our gut microbiota has responded to the industrialized lifestyle, which, evolutionarily speaking, is a very, very recent thing. We've discussed aspects of this lifestyle all day, like the heavy reliance on mass-produced, highly processed industrial foods, common use of antibiotics and Western medicine practices in general, things like cesarean sections. Our habitat, as Jessica talked about, and to which I'll add things like high population densities and relatively low activity levels. This industrialized lifestyle is correlated with certain diseases, and the question is how much of this is due to negative changes to our gut microbiota brought about by this lifestyle. Now, I came to this question through an unconventional means. I've been looking at the diets of our ancient ancestors, everything from Neanderthals to Australopiths. And to understand the diets of these ancient populations, we use a system of proxies, from looking at the bones of the animals that they ate or the microscopic residues of the plants they consumed. But unfortunately, there's no convenient proxy for understanding the gut microbiota of extinct species. It just doesn't preserve in the historic or archaeological record. And it's really not even possible to directly query the gut microbiota of modern people living 50 or 100 years ago. So the only option that we have really is to study people who live in a manner, what we think of as the undisturbed state. In other words, to look for other living populations who don't have industrialized lifestyle and try to draw meaning from the similarities and differences that we see. Only a handful of these traditionally living groups have been studied so far, and yet they show some surprising similarities, despite really broad geographic range, groups from Central Asia, Sub-Saharan Africa, South America. And despite that they live a variety of different lifestyles, some are subsistence farmers, some are semi-nomadic pastoralists, and the group that I worked with are hunter-gatherers. As Justin already mentioned, the first and likely most important similarity among these groups is that they have much greater diversity in the microbiota. Some other important things, though, like industrialized populations, their gut microbiota is usually dominated by two particular phyla, Firmicutes and Bacteroidetes. But the traditionally living groups usually have a much larger representation of Bacteroidetes. And the third similarity is that there are several rare taxa that are found in relatively high abundance among traditionally living populations, particularly those groups from Africa that have been looked at so far. These microbia include Prevotella, Succinivibrio, and Treponema, and you might recognize the name Treponema since it's the genus that includes syphilis and yaws. What I want to emphasize today, though, is that despite these three main commonalities among the traditionally living groups, it's difficult to interpret what they mean. All of these groups are modern living people. They do not and cannot directly represent our ancestral state. They have their own evolutionary and cultural history, and they often interact even with industrialized groups. The group my team studied, the hunter-gatherer Hadza from Tanzania, regularly interact with the local pastoralists who bring their cattle into the Hadza land, and up to 5% of the food that the Hadza eat come from trade with the pastoralists, from trade with tourists, or even the researchers themselves. We don't know what the increased diversity of microbiota really means, in part because many of the microbiota found in these studies are just, we can't identify them. Someone mentioned earlier, we just don't have the reference phylogenies to identify these microbes down to the level of genus or species. We also, and this was talked about earlier as well, have limited understanding about the function of many of the taxa within a gut microbial community. We know that the function as a whole is usually conserved, and that sometimes old microbiota species adopt new rules by acquiring novel DNA from the environment. or that new species may quickly sort of arise to fill new niches. But this question, then, what does it mean to have more Prevotella and Treponema than industrialized groups? Do these genera fulfill some core function within the gut microbiota, or are they simply opportunistic colonizers that may adopt new roles aligned to environmental constraints or the behavior of the host? Even this question of the Firmicutes to Bacteroidetes ratio is complicated. Among industrialized populations, a lower Firmicutes to Bacteroidetes ratio is often seen in lean individuals, while obese individuals have a higher ratio. This ratio changes as a result of diet. We've seen among a Hutterite population in Canada, in the summer when they eat more fresh fruit and vegetable, they have more Bacteroidetes. But in the winter, when they eat more preserved foods, they have more Firmicutes. This seems to suggest that the ratio of Firmicutes to Bacteroidetes is a reflection of the diet, and that the increase in Bacteroidetes is an adaptation to a higher fiber, increased plant carbohydrates, the kind of stuff that Justin was just talking about. But this interpretation is contradicted by the study of Mongolian pastoralists, who also have more Bacteroidetes relative to Firmicutes, despite their traditional diet being very high in fermented milk, red meat, alcohol, and low in fiber and other plant carbohydrates. So we really don't know how strongly tied this ratio is to particular dietary components like plant carbohydrates, or if it's instead, if it maybe reflects some other influence. And so based on these inconsistencies and uncertainties, I urge caution when interpreting the meaning of gut microbial patterns among traditional living groups. It's too soon to suggest that the gut microbiota seen in one group, or even shared across several groups, is the ancestral profile or even a healthy profile, given the complex relationships among culture, function, and the evolutionary history of the studied groups. We still have a lot of work to do here and need to study some more groups to try to tease apart these influences.